I am Mrs. Sloan, and this is a video for my AP biologist. This for us is chapter 11, Mendelian Patterns of Inheritance. So I'm going to make myself a little bit smaller over here and go ahead in presentation mode. And I need to start off by just talking to you about, it's 5.3 if you look on the unit guides for AP Bio. And by the way, unit guides are located on my website. And the notes that my students use are down in the descriptor. Column 1, you fill in the notes. I'll help you with that. Column 2 will be pictures and images that help you. All right, and this is going to be video 1. So I need to start off by talking about genetics, heredity, and evolution. And this is 5.3 in the unit guide. And so on that first part, it talks about how you need to be able to support that, and on your notes, genetics, heredity, and evolution, shared, conserved, fundamental processes and features support the concept of common ancestry for all organisms, common ancestry for all organisms. So we've talked about that a little bit already, right? Luca, common ancestor. And so we're going to look at 3 specific pieces of evidence. I keep getting a glare in my eye. 3 specific pieces of evidence to support that and common ancestry. Now, some of what I'm going to talk with you about right now, we haven't got to those chapters yet, but I am going to introduce all the evidence to you now. You know a little bit about it, and then we'll revisit it once we've had discussions over those chapters. So these processes and features provide evidence, this goes in your notes, number 1, evidence that all organisms, archaea, bacteria, and eukarya, both extant and extinct, both living and now extinct, are linked by lines of descent from common ancestry, linked by lines of descent from common ancestors. So first piece of evidence, okay, is just DNA and RNA. So DNA and RNA are found in all cells, one form or another, and even non-living things like viruses, right? That's how they take over host machinery. So on your notes, DNA and RNA are carriers of genetic information. The proper storage and transfer of this information are critical for life to continue at the cell, organism, and species levels. So non-eukaryotic cells, if you look right here, here's a bacteria. Non-eukaryotic cells, the DNA is circular. Let's put that in your notes, circular. Eukaryotic chromosomes are linear, like what you're seeing right here in this karyotype. And you have multiple linear chromosomes in a eukaryotic cell. Point 2, ribosomes are found in all forms of life. And what do you use ribosomes for? Right? It's the workbench for protein synthesis. Viruses, you got a virus right here. Viruses have either RNA or DNA, and it could be either single-stranded or double-stranded. And then DNA is interchangeable between living organisms. It can be moved from one organism to another. You can take a human gene and insert it into a bacterial cell and get that bacterial cell to make human insulin for you. Okay, so that's what we mean by interchangeable. And if you're familiar with the central dogma, which is DNA is our codebook for information. You use that codebook to make a copy of mRNA. Remember, M stands for messenger. And then you use that mRNA to build proteins. Proteins are built out of amino acids. And trust me, we have a couple of chapters just on this right here, so don't stress out too much about it. But I'm telling you how the nucleic acid gets translated into amino acid language of proteins. So DNA codes for our proteins. And that is what's called the central dogma. Now, every 3 bases that you see right here on the mRNA is called a codon. And there is a chart right here, the universal genetic code. And it doesn't matter what kind of organism you're in, those codons speak the same language in all cells. So a particular amino acid, like CCU, will code for the amino acid proline. So it is a universal language. So B, a universal genetic code. A, T, G, C, and U are nitrogenous bases found in all living organisms. The central dogma of genetics states that in all life, this is a conserved mechanism. So DNA replication ensures the continuity of hereditary information. So you know about DNA replication, right? Because that's going to happen right before mitosis and meiosis. DNA replication ensures the continuity of hereditary information. Number 2, genetic information flows from the nucleotides, and I'll go right back here, flows from the nucleotides in a gene to a sequence of amino acids, in the protein. And going from DNA to RNA is transcription, and using this RNA to build a protein is translation. And then phenotypes, what an organism looks like, are determined through protein activities. All right, so second piece of evidence is the universal genetic code. Third piece of evidence is metabolic pathways, okay? Who does glycolysis? All cells. If all cells do that, then the last universal common ancestor did that. That supports the idea that we came from this organism. Why we all use DNA. We all use RNA. We all use the universal genetic code. We all use ATP for energy. So this, and along with signal transduction pathways that are similar, right? You have the receptor of that signal transduction, and then some sort of response that supports that we have common ancestry. So underneath metabolic pathways, ATP energy, glycolysis, and either aerobic or anaerobic respiration. So the word you need is glycolysis. Aerobic or anaerobic respiration to make ATP. Cell communication in all organisms involves transduction of a stimulatory or inhibitory signals from cells, organisms to the environment, right? To know what to do and when to do it. And so we studied that all last unit was about signal transduction pathways. And when you have a single cell, it influences how that cell is going to respond. If you're a multicellular organism, it coordinates activities within that whole organism. So on your notes, single-celled organisms, signal transduction pathways influence how the cell responds to the environment. And then multicellular organisms support signal transduction pathways, coordinate the activities within individuals that support the function of the organism, as a whole. So let's look at a couple of examples of that. One is quorum sensing. This is a regulatory system that allows bacteria to share information with other bacteria and with themselves to control what genes are expressed. So if there's overcrowding, they might express a different DNA than if there's plenty of room. And so you can see some of the responses here are group behaviors, bioluminescence, virulent, if they're going to become pathogenic, pathogenicity, if that's going to increase. All right, so that is there. Then in multicellular organisms, we've already studied this. We've talked about paracrine signaling, local signaling, right? We've talked about signaling at the synapse. We've talked about signaling as a distance within an organism with hormones. And then we even talked about pheromones between two different organisms. So this process of cell signaling is a pathway that all cells do. So the existence of these properties in organisms today implies that they were present in a universal ancestor, and that present life probably evolved from that ancestor. So here, shared conserved fundamental processes and features support the concept of common ancestry for all organisms. What are those shared processes? DNA and RNA and ribosomes, universal genetic code, and shared metabolic pathways, as well as signal transduction pathways. Now, in eukaryotes, we have some similarities as well, right? The cytoskeleton, the nucleus, membrane-bound organelles, linear chromosomes, our endomembrane system. That's a universalness of all eukaryotes. So in eukaryotes, conserved core elements provide evidence for evolution. The presence of a cytoskeleton, a nucleus, membrane-bound organelles, linear chromosomes, and the endomembrane system. Now, talking about that DNA, if you were to recite the order of all the bases in your DNA starting tomorrow morning, okay, and you listed off those bases in your DNA, in your cells, and you did 100 a minute, it would take you 57 years to say all your bases. And that is if you didn't eat, sleep, go to the bathroom, nothing. Okay, 57 years. That's how many bases you have in your human genome. And so what we're attempting to do in this chapter is to talk about how do we pass that information, that huge amount of information, down to our offspring, and how does that DNA code for our actual traits. And we will look at the connection between the genes and the traits, and then in a further unit, we will actually talk about the biochemistry of that process. So in order to do that, we need to talk about the father of genetics, and the father of genetics, his name is Mendel. Gregor Mendel died without knowing he was the father of genetics. His work was left unread by anyone of significance. If Darwin would have seen it, it would have been so helpful for him because they were contemporaries. So he was an Austrian monk. He studied math and science at the University of Vienna. So here is the University of Vienna and their botanical gardens. He was a substitute teacher. He had farmers for parents. His parents were farmers. And that could explain why he felt so comfortable in his monastery growing peas and doing work with peas. His work went unrecognized during his and Darwin's lifetime. Before Mendel, there was what was called the blending concept of inheritance, which was kind of a mixture. So they would say that if there were, yellow peas and green peas, then you would just mix those colors together, and then the offspring would be some kind of blending, some color in the middle. Now, this blending concept, no help to Darwin. It didn't account for why you had specific variations and why those variations then could be selected for and be better, have better adaptations for that particular environment. Darwin needed to understand that part because having variations was so critical to his theory of natural selection. But due to Mendel, the particulate theory of inheritance came about, and it could explain the differences and variations in those offspring. So traits, let's see, on your notes, sorry about that. Before Mendel, blending, offspring would have traits intermediate between those of the parents. It did not help Darwin because it did not account for variations. So what did he do with this? He worked with peas. He worked with garden peas. He crossed them and crossed them and crossed them. And he crossed so many. He kept such careful records. He used statistics, which is why math is so important to science. He prepared his experiments carefully, conducted preliminary studies. He worked with the garden pea because it was so easy to cultivate. It had a short generation time, and he could cross-pollinate because they had both sexes, right? He could cross-pollinate. He chose 22 true breeding varieties, and I'll go over some of those with you, like a true breeding tall plant, meaning that it always made tall offspring, and true breeding short plants, all of its offspring were short, and then he would cross those and say, hey, what came out? Because if it was blending, if you crossed a true breeding tall pea plant with a true breeding short pea plant, then you would assume if there was a blending form of inheritance, which was what was previously thought, then you should be a middle height, right? But it wasn't like that. The offspring were either tall or they were short, and they were at a specific ratio. And we'll talk about that. Okay, and he kept very careful records. So he developed what's called the particulate theory of inheritance based on existence of minute particles or hereditary units that you and I now call genes. He called them factors, but you and I will call those genes and specifically alleles. So on your notes, number 2, now we would call those particles genes. So on his experiment, Mendel and the garden peas, he used statistics, careful and deliberate in following the scientific method, kept detailed, accurate records. You have the rest there, conducted preliminary studies. He used garden peas. Why? And on your notes, jot down easy to cultivate, short generation time, and can be cross-pollinated. It's right here. Pause if you need to. And he chose 22 true breeding varieties. So he would do reciprocal crosses and then look to see what he got in those offspring. I'll show you those in just a minute. Let me show you some of the traits he looked at. He would cross tall plants with short plants, and tall was always dominant over short. He crossed round peas, full round with kind of shrivelly wrinkled peas, and round was always dominant over wrinkled. And he would cross yellow peas with green peas, and believe it or not, yellow peas are dominant over green peas. Now, when we'll talk about F1 and F2s in just a minute. Now, here are some of the other traits that you could look at. You can look at purple versus white, axial buds versus terminal buds. Now, on your notes, go down to his monohybrid cross. Mono means one, so that means he was just looking at one trait at a time. Later, I'm going to show you some dihybrid crosses. The parental generation, and let's say he was crossing a tall versus short, all the offspring would turn out tall, and you have that on your notes. So he would cross a true breeding tall with a true breeding short, and all the offspring would turn out to be tall. He would take those offspring, the F1s, and he would cross-pollinate them with each other to create F2s. So those would be like the grandchildren, would be the F2s, okay, from the original parent plants. In the F2s, when he would do those crosses, he would get 3 tall ratio to 1 short. 3 tall to 1 short. And here you can see some of his actual numbers. Okay, so if he crossed a true breeding yellow with a true breeding green, all the offspring would turn out, all the F1s would be yellow. But when he crossed those F1s together, then lo and behold, the green traits started popping back up in a 3 to 1 ratio. And you can see all of those here. So why is that? Well, first of all, when he did his reciprocal crosses, he would take the pollen of plant X onto the stigma of plant Y, and then vice versa. And then he would look at all the offspring that they generated. So here, let's look at this. Here's the original parent plant. This one had a wrinkled, smooth, was dominant over wrinkled, but he would cross a true breeding plant with smooth peas with a true breeding plant with wrinkled peas. And when he would cross-pollinate them, the children generation, as it's labeled here, or the F1, they were all smooth every single time. But when he would crossbreed smooth peas with each other and get the F2, the grandchildren generation, it would show up when you would count all the peas about 3 to 1, smooth to wrinkled, or another name for smooth is round to wrinkled. So what was going on there? Well, when you have a true breeding tall plant, the only allele it can give is a big T tall allele. That's all it has. Big T, both alleles. Remember, it's a diploid organism, so it has 2 big T's. That is all it has to give is big T. Well, a short plant, what you do for the recessive trait is you use a smaller letter, little t. Okay, so 2 little t's would give you a short plant. That is all they had to give was a little t. But now in your F1 generation, what you have is a hybrid. They all appear tall because tall is dominant over short, but they're carrying this short allele. So if you did a cross where you had big T, little t cross with another big T, little t, so you could pause right now and work this out on a piece of paper real quick, okay? So you put a big T here. This is a Punnett square. Basically what you do on a Punnett square is you put the gametes on the edges of the Punnett square. So typically the female's gametes go here on the top, and by convention the male gametes go over here on the side. So this female gamete, she can either give a big T or she can give a little t. So you put a big T here and a little t here, separate out our gametes. Remember in meiosis, law of segregation, right? And then you do the same thing here for the male. And then what you put in the center right here are all their possible offspring. It doesn't mean they're going to have 4 offspring. They could have 4,000 offspring. But what are all the possibilities? What could they create? You're putting it here by lining up each gamete with another potential gamete. Then from that, you get a genotype ratio. Genotype is what are the alleles. For instance, in this case, the alleles are big T, little t. That's the genotype, the actual genes. And the phenotype ratio is what it looks like. What does a big T, little t look like? It looks tall. So let me show you what that would look like. Okay, so here I separated out, let's say this is the female right here, big T, little t, and I separated out the male gametes, big T, little t. Why? Law of segregation would tell me to do that. Now, if this egg and this sperm get together, then you have big T, big T. The genotype is big T. The phenotype is what it looks like, tall. Okay, here, if this egg and this sperm they meet up in fertilization. You have big T, little t. By convention, you write the capital letter first. Okay, so the genotype is big T, little t, but the phenotype is the same as this other offspring. It would appear tall. And you can see that happened here. And then in this one right here, you have two little t's together. This is the only one that would be short. Its genotype is little t, and that would make a phenotype of a short plant. So here I wrote the genotype ratio. 1 big T, 2 big T, little t's, and 1 little t. This is the genotype ratio, 1 to 2 to 1. The phenotype ratio is different because all three of these are tall and only one is short, so that the phenotype ratio would be 3 to 1. Now, I'm going to give you some more notes on that a little bit farther here, but I wanted to introduce you to the Punnett square. All right, so let's talk about now Mendel's laws. So there's 3 laws: law of dominance, law of segregation, and law of independent assortment. And I want to remind you right now, he came up with these 3 laws without understanding mitosis or meiosis. He had never seen a chromosome through a microscope. He did not know the stages: prophase, metaphase, anaphase, telophase. He did not know any of those. He came up with these laws just by his careful experimentation. All right, so let's go ahead and talk about the law of dominance. Law of dominance says that some traits are dominant over other traits. For instance, all 3, well, there's 2 different genotypes here, right? Big Y, and the second genotype is big Y, little y. Either way, these peas are going to be yellow because yellow is dominant over green. And so the only way to get this green genotype is to have 2 little y's. You cannot tell, I'm sure, but I have blue eyes. The only way to get blue eyes is a recessive trait. That means both my mother had to give me the blue eye gene and my father had to give me the blue eye gene in order for me to give me those alleles, right? In order for me to have blue eyes. Because if just one of them would have given me the brown eye gene, I would have had brown eyes. So the law of dominance says that one trait or factor can be dominant over another. And then, for example, tall plants are dominant over short, yellow peas are dominant over green. All right, let's talk about the second one. Law of segregation, you learned about this in meiosis. Remember, in meiosis 1, you separate the homologous pairs, and in meiosis 2, you separate the sister chromatids. So these are a homologous pair, same size, same shape, and code for the same characteristics. Now, they haven't replicated yet. If they had replicated, then you would see 2 of that dark brown and 2 of that yellow, right? Okay, but what happens is in meiosis 1, the homologous pairs separate one from the other. You're not going to have them both going to a gamete, okay? So on your notes, in summary, each individual has 2 factors, which we now call alleles, for each trait. The factors segregate or separate during the formation of gametes. And when does this happen? Do you remember what stage do you separate the homologous pairs? Anaphase 1. Each gamete contains only one factor from each pair of factors, right? Only one from each pair of factors. And then fertilization gives each new individual 2 factors again, right? When the sperm fertilizes the egg, you restore the homologous pairs for each trait. So let's remind ourselves, if we look here, here's our tall plant, here's our true breeding short plant. The only thing this true breeding tall plant is able to give is a big T. That's all it has. It still does meiosis 1, but all of them are big T's. Every single one of these chromatids is a big T, and over here they're all little t. So then this is your F1, big T, little t. Notice here you've separated. This is where you see the law of segregation. When you set up your Punnett squares, you're showing segregation, the law of segregation. Here for the eggs, here for the sperm. Then you restore your homologous pairs when the sperm fertilizes the egg. You restore your homologous pairs. Now, if we look right here, you've seen this before because we talked about it when I was talking to you about meiosis, right? So here's a homologous pair of chromosomes. For this case, this one alleles for purple flowers, and this one is for white flowers. During meiosis 1, remember they replicate, so you have 2 copies of each of these, right? This is still your homologous pair even after they replicate. right here. So meiosis 1 is going to separate the purples from the whites, the homologous pair, and then you're going to separate the sister chromatids in meiosis 2. So alleles are defined as alternate forms of a gene that occur at the same gene locus, specific location, on homologous chromosomes, A dominant allele will mask a recessive allele. There are exceptions to that. I will teach you all about that, okay? When we talk about, let me go a little bit forward here, okay? Here's your law of dominance. Review that real quick. So you've got your parent generation, purple. You've got white, okay? So if this is a true breeding purple plant, it's always going to give the big P. True breeding white plant, it's always going to give a little p. So this is your F1, okay? It is a hybrid. It is a purple plant, but it is carrying the white flower. So when you cross the F1s, the F2s, you would expect about 3 purple to 1 white. Okay, and the law of segregation says you're going to separate the P's from each other, right? So you would be separating the big P's from the little P's, right? That is the law of segregation. And then you would separate the sister chromatids, but these would both be big P's and these would both be little P's. All right, and here, okay, showing you again where the law of segregation takes place. Okay, remember the homologous pairs do synapsis during prophase 1, and you can even do some crossing over there, and we'll talk about the significance of that again. And then in metaphase 1, they line up in the middle, and then in anaphase 1, that's when they separate from one another. Now, keep in mind, these chromatids are all going to be different from each other due to what? Due to that crossing over, which increases that variety. All right, so here you can see that segregation. If you had or a hybrid green plant, okay, 2 of them, and you separated, and this is talking about the shell on the outside that holds the peas. What do they call that? I want to say carapace. I can't remember right this second. I'll put it in the ed puzzle. But when that gets separated, that the green is dominant over the white, okay, for the outer covering. So this is the law of segregation shown here. And then you're restoring the homologous pairs through fertilization. And you would expect 3 green shells to the 1 white. All right, so a dominant, on your notes, a dominant, we are on alleles, letter B. A dominant allele will mask the recessive allele. There are exceptions. In the genotype, a homozygous, homo means same, a homozygous would be like what you see right here. Big G, and little g. Both letters are exactly the same. Okay, so you can either have a homozygous dominant or a homozygous recessive. Both alleles are the same. And then a heterozygote would be, in this example, big G, little g. There's a difference between these alleles. Or in your notes, I also have big T, little t. Okay, so that's the genotype. The phenotype is the appearance of the genotype. Is it green? Is it white? Is it tall? Is it short? What does it look like? Okay, so here again, you can see homologous pair of chromosomes. Here you could see this, right? Big G, little g, right? This would be considered a heterozygote. Every single one of these is heterozygote because they don't have the same letters, right? So we have little and a big, one or the other, okay, for each one of those. All right, so phenotype again is what it looks like. So the phenotype here is blue eyes. I know what the genotype's going to be because the only way you can get that genotype is with 2 little b's. Okay, brown eyes is dominant. This brown-eyed person, I do not know if they are big B, little b brown eyes, heterozygote brown eyes. Another name for that would be hybrid, or are they homozygous for brown eyes, 2 big B's? All right, so another trait you can look at is widow's peak. Okay, so widow's peak is when you ever have, when it comes to a point here, might be a little point, it might be a big point right here, but this individual, this is Lance Armstrong, he has definitely a widow's peak. Now, to be a widow's peak, you could either be big W, or you could be big W, little w. Either way, you're going to have a widow's peak. Here, it's Jamie Foxx, right? He has a straight hairline right across. So this, to have a straight hairline, would be homozygous recessive. The only way he could be that is 2 little w's. All right. So at this point, there are practice problems throughout your book. And I'm going to do this one set of practice problems with you. So this one, I ask you, what are the possible gametes? And then here, I ask you, is this representing a gamete or a genotype? So I'm going to do the first one with you, and then I would recommend that you pause and do the rest yourself before I show you the answers on this particular one. So is this a gamete? I would say no. It is not a gamete, right? Because it's got the homologous pair, right? It's got 2, and all of these do. But it's asking you what could be the possible gamete from this. The only possible gamete this individual could make is a big W, right? That's all they could make because that's all they have to give. When you do the law of segregation with this individual, though, you've got some options. Half the gametes would have a big W and half the gametes would have a little w. Okay, so let's look over here. Is this a gamete or a genotype? This right here would be a gamete because you don't have the homologous pair. You just have the 1D. Okay, this right here would be a genotype because you have the homologous pair, right? All of these are genotypes, and I'm asking you what would be the possible gametes. Now over here, I'm asking you, is this representing a gamete or is it representing a genotype? Okay, hit pause. Pause, pause, pause. Okay, I'm going to show you the answers now. All right, so like we said before, you'd only have a big W here because even when you do this law of segregation, all you are separating are big W's. Here you could have a big W or a little w. Here a big T or a little t. And here the only kind of gametes you could make is a big T. This is a gamete. This is a genotype because you have your homologous pair. This is a gamete, okay? This is also a gamete even though there's more than one letter. Why? There's only one version of the X, only one version of the Y, and only one version of the Z. Okay, that's what makes it a gamete. This right here is a genotype because I have 2 P's, right? So I have the homologous pair. I would need, if you wanted to make D a genotype, I'd need another X, either capital or lowercase, another Y or another Z in order to make it a genotype. All right. Now, hairlines. We already talked about this already. I want to give you some practice problems. What is dominant? We know that widow's peak is dominant over a straight hairline. So now, pause your video for a minute and cross 2 heterozygotes for widow's peak. Do you remember what that would look like? Big W, little w, cross with big W, little w, because they're heterozygotes. And then I would like you to give me the genotype and the phenotype ratios. I want to pause. Okay, I'm going to go over the setup now. The setup you would do is you would be crossing, because I said to cross 2 heterozygotes for widow's peak, it'd be big W, little w, cross with big W, little w. Law of segregation says you separate out the W's, right? So you do that for both the eggs and you do that for the sperm. Now they haven't identified which is which. I don't care. It doesn't make any difference to the offspring here, okay, because it's not a sex-linked trait. So then you would do your cross right here. Pause again and double check your cross. Okay, I'm going to go over the answer with you. Okay, so the law of dominance, both of these are big W's, so definitely they have a widow's peak. Here, this individual is carrying the straight hairline, but the law of dominance says that big W is dominant over little w, so this individual would also have a widow's peak, as well as this individual. The only one who would have the straight hairline would be the 2 little w's. All right, let's do another practice. I would like you to cross a heterozygote for widow's peak with a homozygote recessive. So pause. Okay, I'm going to go over the setup with you now. Heterozygote with a homozygous recessive. So here is your homozygote recessive, 2 little w's. Here is your heterozygote, big W, little w. So you would fill then in your Punnett squares. Pause if you needed help. Okay, and this is what it would look like. So when you do your ratios on these, okay, because I didn't go over the ratios with you. Sorry about that. I'll go back. The ratio here, you won't write 2 to 2. You would write 1 to 1. Okay, you would always simplify it. So my genotype ratios would be 1 big W, little w, to 1 little w, 1 to 1. And my phenotype would also be 1 widow's peak to 1 straight hairline. Let's go back here to this other one. Oh, here we go. When we did this cross, my genotype ratio would be 1 big W to 2 big W to 1 little w. And then my phenotypic ratio is right here. It would be 3 widow's peak to 1 straight hairline. All right. Now, Mendel and the laws of probability. You actually already understand this. If you know how to flip a coin, you know if I flip a coin, I can either get heads or I can get what? Tails. Now, if you and I were both flipping coins, like as we're watching this video, if I flipped a coin and got heads, okay, what are the odds that you would also flip a coin and get heads with me, right? This is the laws of probability. So let's take a look at this. Odds that I got heads, it's 50-50 or 1/2, right? 1/2 that I would get heads. What are the odds that you would get heads with your coin? Also 1/2. So what are the odds that it happens together? You multiply the probabilities together. So 1/2 times 1/2 would be, we had 1/4 of a chance of both of us getting heads. We also conversely had 1/4 of a chance of both of us, you and I, getting tails. What about some sort of head-tail combo? This is when you start to add things up. So if we look right here, okay, so I wrote on this one. So the odds that you and I both, just go read from left to right. I'll show you the colored one in a minute. Reading across, I had 1/2 a chance of getting heads. You had 1/2 a chance of giving heads. We multiply the probabilities of us doing it at the same time. You multiply it together. 1/2 times 1/2 is 1/4. There was a 1 in 4 chance you and I would both get heads. There was a 1/2, 1 in 4 chance for us to get tails, okay? There was only one way for that to work. I had to get heads, you had to get heads. There was only one way for that to happen. Now, heads-tail combo, I get heads, you get tails, or I get tails and you get heads. Since there's 2 different ways to get that, we add up those force together to make 1/2, right? Because I could have gotten heads and you could have gotten tails this way, the yellow one, and that would have been 1/4 of a chance. Or we could have reversed tails, heads, the green way. So since there's 2 different ways to get any kind of difference between us, to get a hybrid between us, that's when you add it up, because there was 2 different ways to get that. So a 1/4 and a 1/4 is 1/2. This should look remarkably like a Punnett square, right? If we made a Punnett square and put heads, tails, right? 1/4 of a chance, heads, tails, tails, heads. Turns out the same way, it's different, and 1/4 of a chance of tails, tails. So on your notes, okay, go to Mendel and the laws of probability. And we're going to apply this to genes in just a little bit. The product rule of probability, okay, the product rule. To find the chance of 2 different events occurring together is the product. You multiply the chance of them occurring independently. So my getting heads, you getting heads, we multiply our independent probabilities, and together it would be 1/4 of a chance. Okay, and I gave you that right there in your notes. Okay, and this is what we're going to use as shortcut for in dihybrid and trihybrid crosses, so we don't have to make punnets. Okay, huge punnets. Okay, then the sum or additive law, okay, that was the product law. This is the additive law or rule of probability, is that if the same event can occur in more than one way, we would get a difference between us and coins, either head, tail, or tail, heads, 2 different ways to get the same results. Then add all the probabilities together. And for example, same parents' chance of having a heterozygote, because it could go either way. Okay, and so we put a pin in that because I'm going to come back to that. Okay, product rule versus the sum or additive rule. The only time you have to use the sum rule is if there's more than one way to get the same thing. More than one way to get the same thing. All right, so freckled parents have non-freckled children. What is everyone's genotype? Okay, and in this case, okay, so we have freckled parents. Freckles are dominant over no freckles. Okay, so these parents all have non-freckled children. How could that be? Well, they each must have been carriers for the non-freckle trait, right? So The dad must have been big F, little f, and the mom was big F, little f. And this first child they had, let's say this is child number 1, they had a 1 in 4 chance. If you do the Punnett square, right, big F, little f, put it all together, they had a 1 in 4 chance that this child would not have any freckles, okay? Now, what you see up here is chance has no memory. When they had their second child, it was also a 1 in 4 chance that they would not have any freckles, right? And when they had this third child with no freckles, it was a 1 in 4 chance, because chance has no memory. It's not going to go, oh, you know, I already had a non-freckled child, so my next 3 kids all have to have freckles. It doesn't work that way. It's just telling you the individual probability for each of those events. Okay, and you have 3 practice problems, and I encourage you to do those 3 practice problems right here, and then I will be going over these in class if you are in my class. All right, chance has no memory. Each offspring has the same probability of inheriting a trait. Each offspring has the same probability of inheriting a trait. It is not dependent on the genetics of the offspring before it. It is not dependent on the genetics of the offspring before it. And then the last thing I want to go over in this video is about a test cross. Test cross is when you are unsure of the genotype of a particular plant or organism or person for that matter. So for instance, you know what color my eyes are, they're blue. So you know what my genotype is, little b, right? But if we had a brown-eyed person, you would not know if they were a brown or brown carrier for blue. So this is where a test cross comes in, and this doesn't really apply to humans very well, but it is kind of funny. So here we have a white flower. That is a recessive trait, so we know it's 2 little p's. Here we have a purple flower. We do not know if this purple flower is big P purple or big P, little p purple. So who would we cross it with to find out? It would not be helpful to cross it with another purple flower, because statistically speaking, they're probably going to have more purple flowers. So it's not going to reveal. We need to cross it with something that will reveal what it has. So what you would do is you would cross it with a homozygous recessive, because we know every single time a homozygous recessive can only give a little p. Just like I have blue eyes, the only thing I could give in all of my eggs are little b's, right? That's all I have to offer. So if you cross this purple plant, and if it happens, we don't know, but if it is big P, and this plant, you know, is only going to give little p's, then what will the flower colors be of the offspring? Okay, hopefully you said they would all be purple, because if this parent plant is big P, it's only going to have the big P purple, and purple is dominant over white. Now, if this parent, though, is a heterozygous purple, okay, big P, little p, and we cross it with a white flower, will the results be the same? And hopefully you're telling me, no, Mrs. Sloan, they would not, because I would expect only half the plants to be purple and half the plants to be white. Okay, so underneath test cross, the purpose is to determine or confirm the genotype of a dominant phenotype. To confirm or to determine the genotype of a dominant phenotype. So if you have a tall pea plant, what should you cross it with if you want to establish if it is big T or big T, little t? If you have a tall pea plant and you don't know if it's big T or big T, little t, who would you cross it with? Hopefully you're telling me a short pea plant, little t, right? I think this is good for now. We'll do this quick review, and we'll do some genetics and some more practice problems in video number 2. So otherwise, I'll see you in class. I hope that was helpful.